Hi, everyone. Welcome to the Physionic podcast. My name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine. I hold my master's in exercise physiology. I've worked in a metabolism laboratory, actually specifically in a muscle metabolism laboratory, and I've actually done some of these measures that we're going to be discussing in this particular paper. The paper is actually brand spanking new. I was actually told about it late last week by another researcher in a lab that's close to the lab that I work in. And the paper is titled Excessive Exercise Training Causes Mitochondrial Functional Impairment and Decreases Glucose Tolerance in Healthy Volunteers. And this is a really, really interesting paper. I'm going to be breaking up the podcast into two different, I think I finally came to the conclusion of two different sections. This first one is going to be touching more on the insulin resistance, as well as some of the mitochondrial changes in metabolism. And the second part is going to be discussing more of the mitochondrial fission, fusion dynamics. Essentially, how do the mitochondria physically react to excess training, excess working out? So overreaching, burning out. Maybe you shouldn't call it burning out, but overreaching, doing more than you could normally sustain. And it's also going to include some measures of mitochondrial health, like reactive oxygen species production, free radical production, things of that nature, as well as a potential mechanism as to why this whole process occurs. So this paper is really interesting because it covers a lot of different areas, some of it well and some of it not so well. I wasn't as completely convinced as the researchers were with the data that they provided, but we'll be getting into that. So the purpose of this study is to investigate how the amount of exercise affects mitochondria dynamics, so kind of the how they behave, and blood sugar levels, as well as a bit on metabolism. So to give us a bit of background, and if you'd like to actually see the visuals, this is probably going to be a pretty long podcast because I have a lot of data to go through. If you'd like to actually see that data, then please hop onto the visual form of this podcast because I do have a lot of stuff that I've put together. So hopefully you guys get a lot out of this because I really took a lot of time to curate all of this information and some of the graphics that I've created for your educational benefit. So bear with me also because I'm going to be definitely going to more of the nitty-gritty on metabolism. In my last few podcasts, I had a few people say, you should go more into the nitty-gritty and not necessarily hold back. So I think that's what I'm going to actually end up doing in this podcast. So if certain things, go over your head, feel free to ask questions in the comments and certainly check out the visual version because I am going to have a particular diagram that I created. It took me like an hour to put this one diagram together. So it'll give you visuals of what I'm talking about. But again, as always, I'm going to try my best to explain things so you don't have to actually see the visuals. Okay, so a bit of background. So the mitochondria are a major source of cellular energy. That's, mitochondria are the powerhouse of the cell. That's what everybody says. It's like robotic. As well as a source of reactive oxygen species, which we're not going to go into in this podcast, but and a reduction in mitochondrial function is associated with increased insulin resistance. So if that's necessarily the cause, they're kind of trying to decide if it's the chicken or the egg kind of scenario, which one kind of comes first, insulin resistance or mitochondrial dysfunction. Now, the association between mitochondrial dysfunction and insulin resistance and the link is attributed not only to mitochondrial numbers, so the actual number of different mitochondria that you have, if you have 1,000 or if you have 1,500, just as arbitrary units, but also the respiratory capacity. So the ability for mitochondria to actually produce, to actually have the function that they're supposed to have. So that is defined as respiratory capacity. That is the ability to use oxygen to produce energy. And I'll go into a little bit of that today, as well as overall morphology. So the look of the mitochondria, the mitochondrial dynamics, which I'll cover in the next podcast. One regulation, and I'm going to talk about this more in the next podcast and actually show you visuals of what this mechanism is. But one of the regulations of mitochondria is through this nuclear factor, this protein. So it's a protein within the cell that's called NRF2. The long version of it is called a nuclear factor E2P45 related factor 2, and it's related protein called KIAP, which is Kelch-like ECH associated protein 1. Hopefully you're okay with me abbreviating with NRF2 and KIAP. So how these two proteins interact is really important when it comes to mitochondrial function. So KIAP actually regulates NRF2, meaning that with NRF2 being responsible for the production of antioxidants, so the actual production of new antioxidants within the cells, mitochondrial biogenesis, so the production of more mitochondria. So that's why they're interested in this. And KIAP actually reduces the effect of NRF2. So it will bind NRF2 and stop it from moving from a particular part of the cell known as the cytosol to the nucleus where all the genes are kept. So with that, then we can go and learn more information based off of that link between these two proteins. But again, I'm not going to be discussing that in this podcast. So if you want more details on that, check out next week's podcast. But the researchers do end up looking at this link. Now, another quick note, and something that's probably not going to be too happy, it certainly wasn't happy for me when I read this one line. They have one sentence about this. Excess exercise, however, can lead to heart abnormalities like calcification. So clearly there is a threshold for exercise where it becomes detrimental. And I knew this in other ways as well. For example, with women, you can get amenorrhea or loss of a menstrual cycle. And you can also get microfractures that occur in the bones and things like that. So yes, we already knew that too much exercise, but what other things happen to the cells when we undergo too much exercise? So briefly, I'm gonna throw up my first visual here again. If you're just listening, don't worry, I'm going to describe things. So just really simply, what we're looking at here is essentially muscle tissue. So the researchers, I'll go into the study design in just a second, but the researchers are going to end up taking some samples from these human beings. So this is all in humans. And then with that, they can run a series of different experiments on that tissue that they end up collecting from these people. And of course, they undergo different levels of exercise. Now, the tissue itself is largely made up of cells, right? So the cells that we're interested in are the muscle cells, so these myocytes, or muscle cells. And the muscle cells have a large network. In the visual, I have them bean-shaped of mitochondria, but really it's a network of mitochondria that are very long and elongated. And that elongation is really necessary because the muscle cells need just mass amounts of ATP. They produce, I think, something like 80 kilograms. I think that may be marathon level, although maybe that's at rest. I don't know. It's mass amounts of ATP, however. And that's what we're interested in. We're interested in what happens to the mitochondria when these individuals undergo varying amounts of exercise. And I wanted to give you kind of an idea of where the mitochondria are. They're inside the muscle cell. So you have a single muscle cell, and then inside that you have a network of mitochondria. That's going to be important because as I discuss some of these metabolic consequences and stuff like that as we go through the podcast, it's going to get pretty detailed. I mean, I can certainly go into more detail, but It's important to know where things are, and I find that always incredibly important. Okay, so the study design. How did they go about this study? The researchers recruited 6 women and 5 men. So yes, they didn't just focus on men in this situation. And these individuals were healthy otherwise. So they underwent training, which was supervised for 4 weeks using high intensity interval training. So we're not talking about like marathon running. We are talking about a type of training that would be a little bit in the middle of, like, some long, it's running, sure, or it's some form of running like activity, but it's far more intense. So you can only do it for maybe a minute or a few minutes. So it is supposed to be more of a little bit like lifting weights, kind of in the middle. So high intensity interval training with increasing difficulty with each week. So the way it worked is that they took samples, they took blood measurements, and they did muscle biopsy. So they took pieces of muscle from these individuals at baseline. And actually, I'll go ahead and throw up this first visual. So at baseline, and then the first actual week, and the baseline is obviously what they're able to compare against. So the first actual week is a light training week. So they only do about 36 minutes for the entire week of high intensity interval training. Then the second week is moderate training. So they do 90 minutes. And then they have their third week. And the third week is the excessive training. So it's overreaching. It's trying to burn out almost. So they go up to over 150 minutes of high intensity interval training. And then from there, the fourth week, the final week, they actually cut back down to what's called kind of a recovery week. So they only do about 50 minutes of high intensity interval training in that recovery week. So then within each week, the researchers did experiments like taking muscle biopsies to measure muscle mitochondria, as well as performing what's called an oral glucose tolerance test, which I'll go into later on. Okay, so that's kind of the breakdown. It's pretty simple, just a 4-week study, but they wanted to ramp up week to week the intensity, and there's certainly far more data to this than what I'm going to be covering here, but I am covering a lot of data. So again, bear with me. Okay, so I'm going to be using a single schematic, a single diagram, and it is overwhelming. So I'm letting you know now, but we will continuously go back to this single image that will represent everything that I'm covering in this paper. So again, bear with me. Okay, so there's a lot here, but the first thing that the researchers ended up looking at was what's called the maximal respiration. So what is that? The maximal respiration is the use of oxygen the use of oxygen, but also all of the substrates that need to be there for the cell to actually function, to actually be able to produce ATP. And that is a measure within what's called oxidative phosphorylation. So oxidative phosphorylation is where we get this oxygen measure. And so that all occurs within the mitochondrion. So they're doing a measure essentially of the mitochondrial capacity, the ability for mitochondria to function. And I'm going to come back again and again to this particular schematic, but oxidative phosphorylation is, you may have heard me talk about this before, but it is the electron transport chain. So it's the ability for complex 4 specifically to use oxygen. And I'm actually going to have some content just as a teaser. I have a series of videos that's going to actually be explaining and talking about why we breathe oxygen, why oxygen is killing us, what we can do about oxygen killing us. So that's kind of a teaser, but it's a cool series of videos that I kind of put together. They'll release, I don't know, in the next month or so. But the point is that this electron transport chain, these 5 proteins, or sometimes people just focus on the 4, these 4 proteins, 4 or 5 proteins, end up using oxygen, and that's where we get this respiration, this cellular respiration that we're concerned with this first piece of data. So the first piece of data is showing that with you've got the baseline, then the light training, then the moderate training, and then excessive training, and then you have the recovery week. So you have 4 weeks, but the baseline is just kind of before the first week started. And what they showed is that there was a dampened mitochondrial respiration or cellular respiration with the excessive training. So this already starts the story of like, okay, well, what's going on here, right? Because this is a measure of mitochondrial function, or it's a type of measure for mitochondrial function. So you see that if you're actually seeing the visuals, that the comparison to ET, which is excessive training, that you see a dampened response, and you don't see a full recovery either with the recovery section, or the recovery week, I should say, week 4. So then they were also interested, okay, well, to actually undergo this cellular respiration, you can either go through using sugar, so glucose, or you can use fat molecules. And that's, again, what I've got here shown in this schematic. So you can see that glucose can enter, glucose being sugar, blood sugar, can enter the cell, or fat can enter the cell as well. And then both of them have the ability to end up inside the mitochondrion. And when they enter the mitochondrion, they go through a particular metabolic process called the TCA cycle. You may have also heard it in a biology course as the Krebs cycle, but typically we refer to it as the TCA cycle. And the TCA cycle will then, it's a series of enzymes, a bunch of enzymes that make a bunch of chemical modifications to a fat molecule or to a sugar molecule. At this point, technically, they both get produced into the same molecule known as acetyl-CoA. But the point is that you've got a bunch of enzymes that end up doing a bunch of reactions to those molecules, and then they spit out what's called, we're going to call this generally substrate or fuel. With that substrate or fuel, that gets moved to the electron transport chain, those 5 proteins I was talking to you about earlier, and then you get cellular respiration. So this is a measure of, well, what happens, what's going on to fat metabolism specifically. So they're ignoring the sugar part, and they're just looking at the fat metabolism. So with fat metabolism, and this is in a resting state, so this is not while they're exercising or anything like that. So if you look at the graphs, you might think that the excessive training has a lowered fat metabolism. And it turns out that that's actually not the case. According to the statistics, and that's what we really have to base things off, there's no differences between the different groups. So, although I will admit, I mean, it certainly seems like the excessive exercise does seem to be lower, especially if you look at the error bars. It does seem like it's lower. And it may actually be lower. Well, I don't know. Again, according to the statistics, apparently not. But maybe if they'd had a greater sample size, maybe if they'd had 2 or 3 or 4 more individuals that were part of this study, then they maybe would have seen a difference. And just to be clear, it's not like they separated out all of these individuals into separate groups, one group doing light training or anything like that. All of them underwent every single phase of the exercise. So just to be clear about that. Okay, so the next measure, so right, we've got dampened respiratory, we've got dampened oxygen use, or the ability for the cells to respire, if that's even a word. And Then we've got fat oxidation, so the utilization, the use of fat. So with fat, there was no difference, but with mitochondrial respiration, there was a difference. There was a dampened effect with the excessive training. Now they're going to look at another section of the mitochondrium, which is called citrate synthase. Now, citrate synthase is an enzyme that is found in the TCA cycle. So that TCA cycle, which I just talked about, that both fat molecules and sugar molecules can both enter this particular TCA cycle. Now, the TCA cycle with this citrate synthase, once you've got these molecules, if that's sugar, if that's fat, and that enzyme converts acetyl-CoA and oxaloacetate, but we don't really care about that, acetyl-CoA into citrate. And really what this measure is, it's not really telling you a whole lot of information about, metabolism or anything like that, but it is giving you kind of points that the researchers can start to figure out, okay, well, where is there this reduction in respiration, in mitochondrial function that we saw from the first piece of data? And if you see a dampened effect here, if you see a dampened effect before the production of these substrates to the electron transport chain, then that would at least offer some evidence that there's something wrong in the TCA cycle, or maybe something down or upstream from the TCA cycle. However, when they actually ended up looking at this and looking at the data itself, they looked at two different things. They looked at the citrate synthase protein, because the enzyme is a protein. So they measured the amount of protein, and they found there was no differences across any of the conditions in terms of the protein content. If that was from light training, moderate training, excessive training, or recovery, or even by comparison to baseline, there were no differences. However, the citrate synthase activity was increased specifically by comparison to the excess training. So the excess training had a little bit higher, not massive, but a little bit higher citrate synthase activity. So this kind of gives us some more clues. What's going on here? Again, mitochondrial respiration is down, but citrate synthase activity is at the very least not affected. So we know that, okay? And this also tells us some information about mitochondrial content, not only the function, but also the content itself. Do we have less mitochondria? Because one reason why you might have reduced respiration is, well, you've got less mitochondria, that something is going on. Something has, I don't want to say killed off the mitochondria, but we'll just go with that for layman's terms, that the excess training has killed off the mitochondria, and therefore we have reduced respiration. However, they did a bunch of different measures for this. One of them is citrate synthase, which actually, funny enough, citrate synthase is one of the measures that I used for my master's thesis. And they also did measures, which I'm not going to show any of the data for, but they also did measures for OXPHOS, for the oxidative phosphorylation enzymes known as the electron transport chain, what I mentioned earlier. And they found no differences. So clearly, the mitochondrial content does not seem to be affected, at least based off of the data that we've seen so far. Okay, so then they wanted to do, just one more measure, right? So in this final measure, they wanted they'd measured electron transport chain proteins. They've measured the citrate synthase. They've measured the amount of citrate synthase as well as the activity of citrate synthase. And they also wanted to measure the amount of VDAC. VDAC is a pore or a channel that essentially allows the exchange of a bunch of different molecules from the outside of the mitochondria, so inside the cell still, but outside the mitochondrion, to inside the mitochondrion, and has this constant exchange. So, for example, one of the substrates of the glycolysis, so the glucose, the blood sugar, gets turned into pyruvate, and then pyruvate, that molecule, will then move through VDAC and will then enter the TCA cycle. So VDAC is really important for all that stuff. So then they wanted to measure, okay, well, do we see consistent levels of VDAC? And this would just be another example then of, hey, we've got normal mitochondrial content. So looking at that data, then, well, we find something really interesting because and it seems like there's a trend, right? As exercise amount increases, so we've got light training and then moderate training and then excess training. As that increases, VDAC levels increase. So if we didn't have any other mitochondrial measure, we might think, oh, wow, okay, well then excess training is leading to more mitochondria. But as we know from the content of citrate synthase, as well as some of the data I didn't show, that is not the case. At least we can be pretty certain that that's not the case. So it's specific that mitochondria are producing more VDAC. Why? Beats me. If you have any ideas, well, then feel free to shoot them my way. But it's possible. So the mitochondria, you may have a single mitochondrion, and instead of producing more mitochondrion, that single mitochondrion is just producing more of this particular protein, VDAC, and just inserting it into the membrane of the mitochondrion. And that allows, presumably, much faster exchange of these different ions and different molecules and things like that. So it's an interesting finding, but it really, the only thing that we're really interested in here is that mitochondrial content does not change. And we can be reasonably sure that that's the case. So the takeaway so far from the data that I've covered so far is this might imply that reductions in mitochondrial complex protein efficiency, so the electron transport chain efficiency, may be reduced in some way. Since protein levels were not reduced, which I did not show that data, but I did mention it, so it could be that there's a bottleneck that's specific to OXPHOS, oxidative phosphorylation, and because complexes as like VDAC allow for more substrate in, and citrate synthase is highly active. But the reduced respiration, when they ended up measuring oxidative phosphorylation, would imply that everything up to that point, you're producing enough substrate, you're producing enough of everything, but for some reason, those 5 proteins, the electron transport chain, are somehow affected. How? That is a mystery, but we're going to be picking up some more clues overall as we keep going. Okay, so we've talked about mitochondria, right? We've talked about the effect that excess training has on mitochondria, somewhat of a mystery, kind of some divergent results across the board. Now they focus their attention on blood sugar levels. Insulin sensitivity, things of that nature. So if you're interested in that, certainly stick around. All right. So here, again, I'm going to go ahead and show you some of the data, and then I'm going to explain some of the cellular workings again. So here they're measuring, this is through what's called an oral glucose tolerance test, which I mentioned briefly in the introduction, so OGTT. And all they're doing here is having these people fast overnight, then they come into the lab, and then these individuals are fed it's usually a standard amount of sugar, so 75 grams is the typical amount. And then they measure over, let's say, 3 hours or 4 hours, what happens to their blood sugar levels. And the idea is that, of course, if you consume the same amount of sugar as everyone else, that you would then have your pancreas, which is the part of the body that ends up producing insulin, will start to secrete insulin, and therefore then you have the drive, the cells have this drive to start pushing all the sugar that's just like climbed into your bloodstream. Like your blood sugar is really high at this point because you just ate, which is normal. That's not anything to worry over. But you want your insulin then, if you're sensitive to insulin, to then drive that blood sugar back down. So the sugar gets taken out of the blood and gets moved into the cells. And once it's in the cells, I'll show you what happens from then on. But the point is that in the bloodstream, in the veins and arteries and the vasculature, the blood sugar gets removed. And when you're taking blood measurements, that's exactly what you're measuring. You're measuring what's in the blood. So you want that to eventually reduce. And it's over a standard amount of time. So every person gets measured, let's say over 300 minutes. I think it was 300 minutes, but don't quote me on that. And then they just see, okay, well, how much blood sugar did each person have, or under each condition? So what they found is when they compared the low or the low training or light training to the excess training, or really any of the conditions to excess training, that the excess training had elevated blood sugar levels. Total blood sugar levels were elevated in the excess training group. Then they also wanted to look at insulin levels, and they found that insulin levels were reduced in the excess training group. So that actually raises some really interesting questions or some interesting points in general, because essentially what we're saying is that there's a decreased tolerance for blood sugar. So while blood sugar should be rising, right, and then the pancreas should kick in because it recognizes, hey, we've got elevated blood sugar, therefore you have a bunch of biochemical processes that occur within the beta cells of the pancreas, and they start secreting insulin. And then that insulin's in the bloodstream, it starts binding all these cells like muscle cells, and the muscle cells then open up and allow all the blood sugar in. But clearly what's happening here is that you've got this standard spike in blood sugar levels, and some insulin gets secreted, but not as much as with the light or the basal level, the basal condition, the light training condition, and the recovery condition. So something's going on there, which is pretty intriguing. Now, they did some more measures, however, of that. But actually, I'll cover those, and then I'll go into some of the cellular workings, if you will. So then they did another measure, which is just an insulin resistance calculation, and they found this. So this is a measure called HOMA-IR, and so this HOMA-IR, which is a homeostatic model of insulin, it simply is just a calculation based off of the insulin levels and blood sugar levels, et cetera, et cetera. And interestingly, they found that there was no indication of insulin resistance in the measures that they have. Now, keep in mind that this is at a particular time point. I think all of these calculations were done. We know that all these calculations have been done over, this is a 1-week period of excess training, right? So a lot of this stuff is we're taking a snapshot and we're seeing, okay, well, there's no negative effect. However, if that necessarily translates over time, that's really the bigger question. That's probably something that they're going to end up investigating in the future. But ultimately, based on this measure, that insulin resistance is not a factor when it comes to excess training, even though blood sugar levels were higher and insulin levels were lower. And they actually ended up measuring, I don't have the data for this to post up, but they also did a similar measure of beta cells. So they're not actually taking out beta cells and measuring them, but they're doing a similar kind of calculation called a HOMA beta. And with this HOMA beta test, they're able to essentially determine how much insulin is secreted from the beta cells. And they ultimately calculated that the beta cells were secreting less insulin, which makes sense because the overall insulin levels were much lower. Alternatively, I guess you could also say that the insulin levels were being degraded faster, excreted faster, which is also a possibility. But based off of this beta test, you're specifically measuring the beta cells. Okay, so that's kind of where insulin resistance is and all that, but I'm going to go into even more depth with this. So let me go back to my schematic here, and I'd like to kind of teach you or talk to you a little bit about various proteins that control when blood sugar is allowed into the cells. And the key protein that we're interested in here is called GLUT4, and there are other GLUTs, GLUT, not GLUTE. So we're not talking about ass here. We're talking about GLUT, as in GLUT, for the allowance of sugar from outside of the cell into the cell. So this is really, really interesting because you have to have these channels that end up forming in the cell membrane to allow blood sugar in. Blood sugar doesn't just diffuse across the cell membrane just uncontrolled. It needs to have a transporter that allows the blood sugar into the cell. So you need to have enough of these transporters on the cell membrane to essentially create little holes for blood sugar to be removed from the bloodstream and to enter the cells. And when that happens, the only way that can happen is if the regulation of GLUT is reduced. So the inhibitors of GLUT are stopped from inhibiting. So, and before I keep going on that, the only concept I want you to know in your head right now is that GLUT transporters have to move from inside the cell and have to embed themselves into the cell membrane. And once they do that, then that allows this cascade of blood sugar that's really high in the bloodstream to enter the cell. And I'm going to touch on that again in just a little bit. But let's discuss this data that they ended up discovering. Okay. So the data shows, however, that the total GLUT4 content within the muscle cells was higher in the excess training condition, which is really interesting because this might imply that you've got more blood sugar going from the blood into the cells. But we just said that blood sugar was higher in the excess training condition. So that means that it's staying outside of the cell. It's not going into the cell. So, and I'm sure they were probably thinking the same thing. They're like, okay, so total GLUT4 content throughout the entire cell. So if it's on the membrane, if it's inside the cytosol, the inside of the cell, doesn't matter. Wherever it is, we're measuring it. Total GLUT4 does increase with the excess training. However, then they measured specifically how much of it is actually bound to the membrane, the actual cell membrane, which I mentioned is how GLUT4 actually has its purpose. Its actual function is through its movement to the cell membrane. And they found that there were no increases in GLUT4. So no differences across the conditions for GLUT4 that was bound to the membrane, which is really, really cool. And I'll explain that to you in just a little bit as to why that's really interesting. And maybe you can already piece it together for yourself. Now, a final measure that they did was for AS160. Now, AS160 is a protein that actually controls GLUT, what's called translocation. So the ability for GLUT to move from the inside of the cell to the cell membrane. And AS160, and there's a few different styles of thought on this, but I'm just going to go with the one that I read up on and am familiar with. But AS160 will bind GLUT. And AS160 is what's called the GTPase activating protein, and it's bound to GLUT4, and it actually keeps it inactive when it's bound. But then what ends up happening is, and let me show this on the schematic first. What ends up happening, it's on the right side here. So it's bound to GLUT4, and it stops it from translocating, so moving from the cytosol to the cell membrane. And once it's phosphorylated, and it's phosphorylated by another master controlling protein that I'm not going to go into, but the point is that it's phosphorylated, meaning that a phosphate molecule is attached to AS160. And from that attachment of a phosphate then dislocates AS160 from GLUT4. Now GLUT4 is free and can move up to the cell membrane and have its role. So that's what the researchers ended up wanting to look at. They wanted to know, okay, well, we've got elevated GLUT4 levels, but we don't have elevated membrane-bound GLUT4, so it's kind of like we have a bunch of inactive GLUT4. But is that really the case? Do we see potentially elevated levels or changes in the levels of AS160? And what they found is that no, technically, statistically, no. But again, if you're looking at the bar graphs, it does seem like there's kind of a trend there starting to go up. And if these people had done another week of excess training, you would have seen an increase in AS160 levels. So, and they also did measures for phosphorylated AS160, and they again found no differences. So this essentially starts to, well, I'll go into what this actually means in just a little bit. But the last piece of data on this particular section is to look at fat metabolism. Again, we're looking at the use of fat instead of glucose, instead of sugar, for energy. And that again goes through oxidative phosphorylation. I'm going to show you the graphic again so you can see it. So you've got the fat molecule on the left, and it's going all the way through. I didn't show any of the channels and all that. It usually would go through a fatty acid transporter and CPT and a bunch of other stuff. But the point is that it goes through the cell membrane with help through facilitated, and then it goes through the mitochondrial membrane, and then it enters the TCA cycle, just like I described earlier. And then from there, those substrates that pop off the TCA cycle will then go to the electron transport chain and be used to generate energy. So oxidative phosphorylation. And so that's what they're measuring, but they're not just measuring it just kind of, at a resting state. They're measuring it during exercise, but not in the high intensity exercise at steady state exercise. So they put these people to work. They had to exercise at a particular level or a particular intensity, which wasn't super intense, but it was intense enough to get their heart rate going and whatnot. So, and then they did measures of fat oxidation, or the use of fat utilization. And what they found is somewhat predictable because this isn't super intense. So when it's not super intense and it's manageable, the cells do switch to or tend to use a decent amount of fat metabolism as they can spare it. So that's exactly what we see. We see a ramping up. So with light training, there's a little bit of an increase. Then MT1 and MT2 are just two different measures of this moderate training. And then you've got excessive training. And as the training intensity or the amount of training increases, there is an increase in fat metabolism. So this shows that there is a reliance on fat metabolism, and this actually corroborates a lot of what we've already talked about with increases in GLUT, but not any increases in GLUT in the actual cell membrane. So what does that all mean then, right? So the takeaway here is that blood sugar is elevated after excessive exercise, but insulin sensitivity seems to be normal. So there's no increase in insulin resistance. And again, that's all based off of a snapshot. So if somebody were to do this, I don't know, for like 6 months, would they maybe have issues? Yeah, maybe. Who knows? So fat oxidation is increased at a steady workload. So this might imply a greater reliance on fat metabolism over, blood sugar. And it seems as if the muscle cells, and this is what I was trying to get, I was kind of like hinting at, what could this mean with the whole GLUT levels and stuff like that. It seems that the muscle cells are primed for glucose metabolism by increasing GLUT4 levels, but keeping them inactive. Because we don't see that translocation to the cell membrane. So this might also imply that reduced insulin secretion in an elevated blood glucose environment. If this leads to a negative outcome because well, we're not getting much of an insulin secretion from the beta cells, even though the same amount of blood sugar is in the bloodstream. So the researchers mentioned that they think that that's a negative outcome, that's an unhealthy outcome. I'm not that convinced yet. I mean, I just don't think that based off of this one snapshot that we can suddenly leap to this conclusion that suddenly it's unhealthy. There could be other mechanisms that kind of play a role in here that are exercise specific, that, you know, if you take the snapshot, it may look not that great, although not all the data was bad, right? Insulin resistance was not elevated in the excess training condition. You know, maybe there are some adaptations that occur later on, you know, you never know. So I don't think we can jump to the conclusion that the researchers make. And finally, this is the last bit of data that I wanted to go over, and this is probably the most metabolism heavy section, but looking at blood sugar levels during exercise. So here they're looking at specifically blood sugar levels during the intense exercise. So during the high intensity interval training. So of course, as remember, we're looking at blood sugar levels. So that means that if you have a reduction in blood sugar levels, where is it going? It's not disappearing. It's not going poof and just like disappearing out in thin air. It's entering your cells. So, and this makes complete sense. If you look at the data, right, with light training, you tend to have, pretty high blood sugar levels still, not bad high, like just normal levels. And then with moderate training, you have less and less. And then as you get to excess training, which is the most amount of work that you end up doing, then of course it's going to be at the lowest levels it possibly can be. So that's not a huge shock. Now the question is though, once the sugar goes from the bloodstream into the cell, what happens to it? And this is where we get some really surprising results. I'm going to show you all the data in one graphic, and then I'll explain the mechanism. So they measure glycogen levels. So glycogen is a stored form of blood sugar, or sugar in general. So it takes sugar and ends up turning it into chains of sugars known as glycogen. And this was actually pretty surprising because they measured glycogen levels in the muscles, and they found that glycogen levels were actually higher in the excess training condition compared to the light training condition. So the light training condition actually had the lowest levels of glycogen, which is really, really interesting and kind of puzzling until you start looking at the other two pieces of data. So the other thing that they looked at was an enzyme called hexokinase, and I'll show you what this looks like in just a second. But hexokinase is the enzyme that actually traps glucose into the cell. And again, I'll show you that, and I'll explain it a little bit more in just a second. And then glycogen synthase is another enzyme, and you can just look at the words, right? Glycogen and synthase. So it produces glycogen. So that enzyme is responsible for the production of glycogen. And we see that while glycogen levels are higher, or maybe I should say they're lower in the light training condition, and they're higher in the more intense conditions, hexokinase is elevated in only the excess training condition, and glycogen synthase is also elevated in the excess training condition. Now, interestingly, it's also somewhat elevated in the light training condition. So we're getting a little bit of divergent data here, but when we focus on the excess training, it does make a decent amount of sense. You have increases in hexokinase, and more trapping of that glucose once it enters the cells. And then the question is, okay, well, where is it going? What's it doing? And it's going to the production of glycogen. Now, this might corroborate some of the data that we looked at with fat oxidation. So if the cells are switching over to using more fat, then they can spare the use of glucose to be turned into glycogen later on. But that is a bit of speculation on my end. I mean, technically that is the mechanism, but if that ends up being the case, we definitely need a lot more data. You could like label glucose, for example, to be able to figure some of this stuff out. So let me explain this a little bit more. The blood sugar enters the cell, right? So the GLUT4 you've got blood sugar in the blood, right? And then you've got GLUT4 that translocates to cell membrane. That allows the blood sugar to enter the cell. Now it's just sugar or glucose. And then from there, you have hexokinase, which is the enzyme, will phosphorylate, will slap a phosphate onto the glucose molecule and turn it into what's called glucose 6-phosphate. And then glucose 6-phosphate has a bunch of different potential avenues that it can go. It can go through glycolysis and be used for ATP production. Or it can go through glycolysis and be used for oxidative phosphorylation. So to enter the mitochondria and go through the TCA cycle and go through OXPHOS and then produce ATP. Or it can be used for glycogen production, which is where that glycogen synthase will then eventually turn glucose 6-phosphate into UDP glucose and essentially store it away as glycogen. So that's what we ultimately end up finding out, right? That at least that we have elevated levels of glycogen synthase and we have elevated levels of glycogen. So, that's pretty good evidence there. Now, one other piece of data that I did not actually show is that they also wanted to measure lactate levels. So to go into that a little bit, when glucose enters the cell and it gets that phosphate slapped onto it by hexokinase, if you don't have enough oxygen present, if you're not breathing enough oxygen in and oxygen isn't being delivered to your cells fast enough, your cells switch from what's called an aerobic glycolysis, so oxygen-dependent glycolysis. Glycolysis is a metabolic pathway that breaks up the glucose molecule and eventually either spits out one of two molecules. It spits out pyruvate, and pyruvate will then enter the mitochondria and will go through the ETC, the oxidative phosphorylation. Or if you don't have enough oxygen, because oxygen is being used at complex 4 of the electron transport chain, if you don't have enough of that oxygen there, then it creates a backup. The whole system kind of backs up. It's going as fast as it possibly can, but it just can't produce enough. It just doesn't have enough substrate there. It doesn't have enough oxygen to be able to run through this whole process. So what's the cell going to do? It can't just have this like backup of, just like a bunch of semi trucks loads of glucose or pyruvate at this point. What it does is it converts it to with an enzyme called lactate dehydrogenase and turns it into lactate. So then lactate will then be shuttled out of the cell and will actually go through what's called the Cori cycle. I'm not going to go into that, but the point is that it is an indication, the big picture here is that the production of lactate is an indication that you are going through anaerobic glycolysis, which is oxygen missing glycolysis. So with an independent metabolic pathway that is independent of oxygen. And so if you have elevated lactate, what would you think, right? You would think that the cells are switching more over to the anaerobic glycolysis. However, the data showed that with the excess training, there was a reduction in lactate. Why does that make sense? It makes sense because of the data that we've already gone over, that you get more glycogen production. It seems like the cells clearly aren't, not that they're thinking about this stuff, but they're clearly not too concerned about the use of glucose in this situation for to feed the cells, to actually create this energy demand. So likely they are relying a lot more on fat metabolism here on the left, as opposed to glucose. And then whatever glucose is coming in, they're just shuttling it to glycogen production. Now, it is strange. That is kind of a strange situation because with excess training, I mean, you would think that it would be so much more focused on glucose utilization, but maybe that plays into this pathology, that maybe there is something wrong there, that it has to focus on an alternative pathway to produce energy. And does it have potentially reduced capacity to create energy based off of the very first piece of data that we looked at? Who knows? So the takeaway here is that blood glucose levels decrease during exercise, and keep in mind this is during the high intensity interval training, so not the steady state. So this implies that blood sugar is entering the cells. But what is happening to that blood sugar once it enters the cells? Well, less is going through glucose metabolism, so glycolysis, and more may actually be stored as glycogen. Okay, so that was a lot. I told you it was going to be a lot, and I even now I haven't gone as nerd-like into this stuff as I possibly could have, but certainly more than I have in the past. So, what's the conclusion here? What's the what kind of takeaways? I always find it funny when I look at the graphs, like on YouTube or like looking at the podcast, and I see like, some people are really interested in learning all the mechanisms, looking at the data and all that stuff. And then I have this sudden spike in my viewership, specifically at the point where I put conclusions. So there's a good amount of people that don't care about any of this stuff. They just want the answer. And I don't blame you. I mean, that's cool. But I always chuckle when I see that. It's like a flat line and then suddenly a spike as people tune in specifically for the takeaways. All right. Anyway, what are the conclusions? What are the takeaways? So there's a few different things. Excessive exercise could be leading to a bottleneck in mitochondrial respiration. We did see that earlier, around oxidative phosphorylation complexes, that electron transport chain. Exactly how, however, is unknown. And other hypotheses may also be in play that I haven't mentioned because I haven't thought of them. So I'm always open to different ideas. Excessive exercise also increases overall blood sugar levels. So we saw that with the measure of blood sugar and then insulin right after in the short term. It's possible that excessive exercise primes the muscle cells for blood sugar intake because we saw an increase in GLUT4 amount, but not an increase in that movement to the cell membrane, but may tend to produce glycogen with that intake of blood sugar. During intense exercise, blood sugar drops, implying a reliance on glucose metabolism during just those times specifically. But during steady state exercise, after excessive exercise, the muscles use more fat metabolism. And that's where we get that switch of like production of more glycogen, but we're therefore relying more on that fat metabolism. So there are some clues to some potentially worrisome issues. So the decreased insulin secretion to a blood sugar spike, for example, as well as reduced mitochondrial respiration. Not really sure what's going on there. We need more data, more ideas and stuff to probe. But otherwise, changes don't raise like all too many alarms otherwise. So in the next podcast, like I mentioned, we're going to be discussing mitochondrial dynamics, how mitochondria behave, fission. Fission is the separation of mitochondria. Fusion is the reintegration of mitochondria, which is some of actually my research that I do in the lab, as well as the health of the muscle cells and the mitochondria therein. So is there more free radical generation, so reactive oxygen species, so damaging molecules? And I'll also be covering something that I briefly mentioned at the beginning of this podcast called the NRF2 pathway mentioned in the introduction. Okay, so I realize this was a ton of information, but hopefully if you're listening, you're still able to follow along. And if you're watching the podcast, then hopefully all the visuals helped. I definitely skipped over about half of the data because it's a massive paper. It's huge, but I kind of went over some of the highlights. Okay, well, thanks for tuning in. I really appreciate it, and I will catch you in the next one. See ya.